Question 3. And we've got Spirogyra. And this is an autotrophic organism that inhabits fresh water. So you might have seen one of these on our open day if you look down a microscope. Organelles A and B. So A is this kind of blob there. So A is a blob in a cell. It's probably going to be a nucleus. B, these are our green, I don't know whether you're going to get a green one or not, but these are green little blobs along the cell. These are chloroplasts. The actual width of the cell X and Y is 32.3 micrometers. Calculate the magnification. I'm going to put my fantastic ruler on there and I'm going to measure this as 12 millimeters. So X, Y equals 12 millimeters. So I'm measuring it at 12 millimeters and the actual width in real life is 32.3 micrometers. We're using different units at the moment, so we need to convert them to using the same units. It's easier to convert my millimeters into micrometers. So 12,000 micrometers. So there's 1,000 micrometers in a millimeter. So 1,000 micrometers in a 1 millimeters. And so now I've measured it on the page. So this is my image size is 12,000. My actual size is 32.3. So my magnification is how many actuals go into my image. So the magnification equals my image size, which is 12,000 divided by our 32.3. 12,000 divided by 32.3. And for me that is 371.5. So my magnification, does it tell me what, so I'm going to use a 372 times there. During the day, the concentration of solutes in the ponds changes due to evaporation, so water evaporates off but leaves the salts and things like that. However, the length remains almost constant. What can you conclude about the structure? And it gives you a clue here. Cell wall. Okay, so the structure of the cell wall to survive in different concentrations. 4 marks. Well, we have got. We need to talk about this cell wall being strong and what's going on and how that resists that. So this cell wall, we need to talk about what it's made of, why it's strong, and what's going on in the solution. So cell wall is straight chains of beta glucose, which are cross linked to form microfibrils, and these provide strength to the cell wall. These provide strength to the cell wall. You might have said inelasticity or rigidity possibly. So we're going to squeeze 4 marks in here somehow. So we're saying when the water evaporates, this might, you might get water moving in or out of the cell. So the pond water is becoming more concentrated, and so the water potential is going down. Then water would leave the cell, and you think it might all shrink, but it doesn't. So when solute concentration changes causing water to move. Now, it doesn't really say whether you know, into or out of the cell, and then because it says different solute concentrations, so you know, move out of the cell or you know in the cell, you know, by osmosis. I'd always mention osmosis there. So we've got the idea of water moving in or out by osmosis. The cell wall prevents bursting or you know shrinking. So our evaporation would lead to water going out of the cell, and so that would be you know related to shrinking, or you might have done it the other way around. So make sure if you're giving an example, give the right one. Part D. A lot of blurb here. So this is 3 marks. This is AO2, so relating our diagram and blurb to you know to the knowledge that we know. 2 differences and 1 similarity between these 2 species. So difference. Well, let's do a similarity first. So similarity. The similarity both contain, for example, ribosomes. You could have a membrane. They have DNA. So something like those. Differences. And this is just from your table in your notes, just listing some of the differences. So you know only Spirogyra has, e.g., you know, mitochondria or any membrane-bound organelles, you know, DNA enclosed by your nucleus. Other differences that we could have here. So we've done enough there for the marks. Only Spirogyra, you know, is linear DNA, larger ribosomes. We're just reading from the table in the notes here, but that'll do for us. We only needed 2, and you've got your 11 marks.